Namaste students, Kushro Vidya. I, JVN Dr Chandrakala, Assistant Professor, Vidyapeeth, Women's University, Jaipur. In today's session, we are going to discuss about the cellular totipotency. Totipotency is the ability of a single cell to divide and produce all of the differentiation cells in an organism. In other words, totipotency is the genetic potential of a plant cell to produce the entire plant. Isolated cells from the differentiated tissues are genetically non-dividing and quiescent. To express the totipotency, they undergo dedifferentiation and then redifferentiation. Totus means the entirely and a potent means having the power. The possibility of regeneration and entire plant from a single or a few non-zygotic cells are proposed by Gothenburg Haberland in 1902. Haberland is popularly known as father of plant tissue culture. In 1950, Stewart and his co-workers succeeded in growing carrot plant from isolated phloem cells, where they have taken a fragment of the culture nutrient medium, stirring with the single cell of in the liquid. The single cell freely in a suspended began to divide, and the embryonic plant developed from the culture of the single cell, and the plant was cultured in agar medium. Later, it was planted in the soil. So, a single somatic carrot cells developed into a mature carrot plant. So, here the high ratio of the cytokinin to the cytooxin that has a shoot development, callosogenesis, where the ratio of the cytokinin to auxin is equal to the root development, where auxin is low, then root development takes place, where cytokinin is high. The high ratio of the cytokinin leads to the shoot development, and the low ratio of the cytokinin leads to root development, that is rhizogenesis. Okay. So, when you lower the cytokinin concentration, a root development will take place. As you will increase, the ratio of the auxin is increased, so the root development takes place. So, the totipotency or the differentiation leads to the embryogenesis or organogenesis or development of a cytodifferentiation. So, through embryogenesis, embryoids are formed to develop into a complete plant. Through organogenesis, callosogenesis, or only a shoot is developed, or rhizogenesis, where only root is developed, or callolysogenesis, where shoot and root is developed, or cytodifferentiation, where a xylem or a phloem or any other organs are developed. So the culture system like callus culture, where the callus is unspecialized or unorganized cell growing in a different into a mass of cells, can be maintained indefinitely through a suitability of a cytodifferentiation, where no photosynthetic or which can be grow in dark or so can only use to isolate the single totipotent cells from a callus culture. Many cultures lose their potency or a potential to differentiate during the continuous subculture and forming the epigenic changes, or difficult to follow many cellular events during the growth and developmental phase in a callus culture, uh, where the or unorganized cells has to differentiate and re-differentiate to form organized completely into a plant organ. Or a suspension culture, where the type of culture in which the single cells or a small aggregation of the cells multiplies, which suspend its excitation to the liquid medium or receives homogeneous stimuli. or even it can study in a secondary metabolites such as alkaloids or mutant cell clones from which the mutant plant can be raised. Possible to analyze the biochemical pathway related to the differentiation of the cells, or a single cell culture where methods of growing the isolated single cell aseptically on a nutrient medium under controlled conditions, or a single cell culture is the ideal systems for the study of the biotransformations. Protoplasts are the naked plant cells without the plant cells, but they possess the plasma membrane and all other cellular components. And isolated systems have the most frequent, but the mesophyll types of the fully expanded leaves of the young plant or the new shoots either enzymatically, or the protoplasm is the culture can be regenerated into a whole plant, or a hybrid can be developed into a protoplast fusion. It can perform the single cells or cloning of the protoplasm. So genetically transformation can achieve through genetically engineered protoplast cells. The protoplasm is excellent material for the ultrastructure studies or isolation of cell organelles and the chromosomes easily from the protoplast, or a chloroplast from the which the usefulness of the membrane studied or the transport and uptake of the processes. So some factors that affect cellular totipotency and some of the explants like organs that serve as a tissue source or a physiological or through the seasons or size or overall quality of an explant. Medium nutrient medium, that is organic, inorganic nutrient, carbon source, reduced nitrogen source, or a plant growth hormones can be used, or a cultural environment, presence of a medium, light, temperature, humidity, and other environmental impacts to form here. The importance of that the vegetative propagation for the vegetative propagation or germplasm preservation or the seed dormancy or for embryo rescue or the protoplasm fusion or a haploidy or a genetically modifications here. These are the certain important aspects of having a cellular totipotency. Thank you. This session is powered by 2.0 Vidyapeeth University, Jaipur. If you have any queries, we'll discuss in the problem solving.